Hello, everybody. This seminar presentation is being presented by Dr. Malay for partial fulfillment of refresher course in biological science, excuse me, being organized by UGH-HRDC, Dr. Babasaheb Ambedkar Marathwada University, Aurangbad, Maharashtra. Now, in next 15 minutes or so, I'm briefly going to talk about the bacterial synthesis of nanoparticles. Now, nanotechnology is considered to be a wonder technology of 21st century. It usually deals with nanomolecules, and when you say nanomolecules, those are molecules that are in a size range of 1 to 1000, 1 to 100 nanometers. And 1 nanometer is 1 billionth of a meter. So now the question is a particular molecule of such small, tiny size, why are they be so special, and why there have been so much discussion nowadays being carried out on this particular branch of science, which is known as nanotechnology. It is simply because nanotechnology has immense application in various areas of our daily life that includes their antimicrobial activity, their utility for various cosmetic items, their immense utility in medical biology that includes both medical imaging and in terms of targeted drug delivery, and even nanomolecules can be used as nanobiosensor, for instance, for detection of pathogens in plants. Now, if you want to develop some kind of nanomaterial based technology, first you have to synthesize those nanomaterials. And classically, nanomaterials can be used, synthesized either by using physical methods such as ball milling, thermal evaporation, lithography, and vapor phase deposition, or by employing chemical methods that include salt gel processing, solution based synthesis, co-precipitation method, electrochemical synthetic method, irradiation methods, and tolling methods. However, although all these biochemical and physical methods have been quite popular, but in recent times, the green synthesis or biological synthesis of nanomaterial have become more popular than the conventional biosynthesis methods. And when you say green synthesis, we mean that synthesis of nanomaterial by using any kind of biological organism, be it fungi and yeast or bacteria or even plant and plant extract. And it has already been found that various types of nanomaterial using different metal ions such as gold, cadmium, silver, iron have been used successfully, where scientists have used the biofactory to synthesize the novel nanomaterial. Now, the question is then, what are the major advantages of green synthesis of nanomaterial? There are several advantages, but just to chart a few would be like they are very inexpensive and less cumbersome. In terms of yield, they are quite good because the yield potential is excellent. They definitely require much less energy. They are usually non-toxic, and that means that they do not when those green synthesized nanomaterials are synthesized, they are more biocompatible, and therefore they can be easily used in the medical biotechnology, for medical biotechnology purposes. They do not require the technical expertise. They are much quicker, and the most important thing is that we are fortunate to have so much abundance of microbes in this world that many of those microbes have a huge potential to develop new nanomaterials. Now, out of all these different microbes, as I already have mentioned, which includes fungi, virus, bacteria, why particularly we are focusing about bacteria? Or in other words, why bacteria synthesizes nanoparticle? The major reason is detoxification, because when a bacteria is encounters toxic metal ions, they use its biochemical mechanism to detoxify these metal ions, and as a byproduct, the nanomaterials are being produced, and that nanomaterials we use for various purposes. So when I mean that different ways bacteria can synthesize nanoparticle, this can be categorized in different biochemical ways, such as it can be a modification in the cellular transport machinery to restrict entry or of the toxic material. It can be also a sequestration of the toxic material either inside or outside the cell. It can also involve activation of energy dependent efflux pathway. And I'll show one example where they have nicely shown that it can be even an energy catalyzed oxidation or reduction of the toxic species. So the next question that comes is that what is the general flow, or what is the general procedural way nanoparticle can be synthesized in bacteria? In a nutshell, first the bacteria has to be isolated, and then to that isolated bacterial culture, the corresponding metal ion it has to be added, of which the nanoparticles are required. Then, in order to maintain the culture, specific pH, temperature, and other reaction conditions have to be maintained to allow the proper chelation. Then the question is, after the reaction, how do you know that whether the nanoparticles has been synthesized or not? One quicker way or easier way is to look for color changes. As you see in this picture here, this is the normal, the bacterial color of the bacterial broth being grown in LB media. And once this nanoparticle has been synthesized, the color usually changes, like what is being observed here. And then different standard technique methods can be used, such as SAME, zeta size error TEM, to characterize the synthesized nanomaterials. So, as I already mentioned, that bacteria can be successfully used to synthesize the nanoparticle, and it has already been shown by different research groups that a spectrum of bacteria that includes Pseudomonas, Bacillus, the different species of Bacillus, Listeria, like that, they can be used to synthesize nanoparticle from different metal ions such as silver, cadmium, and then when they synthesize the nanoparticle, they can either use the extracellular machinery or intracellular machinery to synthesize those nanoparticles. This cartoon just briefly shows you as an example that how extracellular synthesis of nanoparticle can be synthesized in case of Rhodopseudomonas by using NADH reductase. Here what happens, first electron is transferred from NADH to NAD plus, and that electron is getting transferred by NADH dependent reductase, and that helps in the formation of this nanoparticle, this gold ion, so gold nanoparticle. So that means that there are biochemical machineries which bacteria use to develop the nanoparticle, but as I already mentioned, they do it in order to just get rid of the toxicity that might have arisen due to the application of the metal ions. Now, the question is, bacteria or other microorganisms, they can synthesize nanoparticle either at the extracellular level or intracellular level. But usually scientists believe that extracellular synthesis is more advantageous than intracellular one. It is simply because if it is synthesized at the extracellular level, then we don't need to break the microbial cells to get the nanoparticles out of those cells. Now, it is, although my talk is mostly focused on the bacterial synthesis of nanoparticle, but I just want to point out that other microorganisms such as fungus and bacteria and virus, they are also capable of synthesizing nanoparticle. And here in this table, it has already been shown that these are different groups of fungi and virus which has so far been found quite effective in doing that. Now, nanoparticle can be synthesized by fungi, as I already have mentioned, and sometimes it is often considered that to be more beneficial than bacteria because of many different reasons. But one simple logic that scientists put forward is simply because of the presence of mycelium, they always offer an increased surface area, and that because simply due to that increased surface area, there is always a possibility that more or higher abundance of nanoparticle or nanomaterial can be obtained if fungi is used as a biological material to synthesize nanoparticle. Now, once we want to synthesize nanoparticle from some metal ion, then for each and every nanoparticle synthesis, there are specific reaction conditions that one has to standardize. And in order to standardize the reaction condition, we need to understand that what are the factors that actually control that how much abundance of nanomaterial will be synthesized, and also the quality of those nanomaterials. So far, the research that has been carried out can be summarized that these are the major contributing factors that include pH, temperature, metal type, and incubation type. These are the major physical and chemical conditions that actually control the quality and quantity of the nanomaterial that is being synthesized after a reaction. And again, in this cartoon, we describe again that what are the different reaction conditions, such as incubation period, mixing ratio, and also temperature, pH, and addition, which I already have indicated in my previous slide. Now, also on different, with respect to different bacteria, scientists have studied the impact of different factors and how they regulate the amount of nanomaterial that is being synthesized. In this picture, I actually describe, or graphically, it is represented that how, in case of Pseudomonas aeruginosa, the temperature, incubation period, and metal concentration, they impact on the amount of nanomaterial that is being synthesized. This data has been obtained from one paper published in Journal of Ecological and Health Environment and was published by Abosted et al. In 2015. If you look at this picture, you'll see the first picture. In the x-axis there are temperatures, and in the y-axis there is absorbance. And if there are higher amount of synthesis of nanomaterial, then the absorbance will go up. So as you move from lower temperature to higher temperature up to 85 degrees centigrade, there is an increase in terms of absorbance, meaning that higher the temperature, the higher the synthesis or abundance of synthesized nanomaterial. But then after 85 degrees centigrade, there is a drop. So this experiment actually proves that in case of Pseudomonas aeruginosa, for a silver nanoparticle synthesis, 85 degrees centigrade temperature is the optimum temperature which actually can produce maximum amount of nanomaterial. Now, when incubation period was considered, the same kind of experiment was carried out here, where the incubation was carried out from 0 minute to 48 hour, and then the data were collected at a regular interval by checking the absorbance. And as you can see, from 0 minute up to 30 minute, there is a steady increase of absorbance, meaning there is a more synthesis of nanomaterial, and then the reaction actually comes to a kind of plateau phase. So that means if you increase the exposure time after 30 minutes, it doesn't matter, but then the abundance of the synthesized nanomaterial will not be changing. Similarly, when metal concentration was also considered, it was found that at a certain specific metal ion concentration or input metal ion concentration, the amount of synthesized nanomaterial was maximum, and then the concentration of the synthesized nanomaterial drops down. Now, in order to conclude the entire talk that I have presented so far, we can conclude in this way is that firstly, green synthesis of nanoparticle is a very environment-friendly method for synthesis of nanoparticle, and bacteria indeed is an ideal biological system for this. Secondly, bio-based nanomaterial synthesis has higher possibility of biocompatibility, and therefore they are less cytotoxic, and that is why they are being highly popular in terms of their drug use, for instance, particularly in cancer industry or for developing anti-cancer drug. Nanomaterial have been quite popular recently. It also shows a higher anti-cancer activity, and that has already been experimentally proven by using different cancer cell lines. It has higher antimicrobial activity, and therefore, quite often these nanomaterials, particularly the nanomaterials which are synthesized by biological organisms such as bacteria or plant or fungi, can be used as an antimicrobial agent. And lastly, the lack of knowledge of the chemical components responsible and the underlying mechanisms for the synthesis, action, and stabilization of biological nanoparticle remain an open challenge. And therefore, what I want to conclude that further research needs to be carried out, since we have so many diverse types of microbes around us and so many different types of metal ions. So more research needs to be carried out to identify more nanomaterials which may have immense application in different areas of our daily life. So with this note, I end my today's talk, and thank you very much for your kind and patient hearing. Thank you.